Hey, what's up, guys? This is Audie from Shastri Nation, and today we're going to be talking about DNA tech. Alright, so now that you're back from the intro, let's go ahead and get started. So, DNA tech. Okay, so DNA tech is basically like the stuff that we use to research, to help us with research, other things like that. These equipment, I guess you can call it, just makes our lives easier, or scientists' lives easier, by helping them out, making molecules and inserting DNA and things like that. So we're going to talk about a few of the major technology that we have right now. So this video is actually going to be pretty short, maybe about 10 minutes, I think, because depending on how much I talk, but maybe about 10 minutes, because really it's just me listing out these things and then saying what they do. Yeah, that's basically it. Now, I am covering E3, so this is, a lot of work for me right now. I have my website up and running, so you can go ahead and check that out at www.shastrination.com. It's the same spelling and everything. I think I'll have it listed in the description too, but I have all my E3 stuff on there, and then I have my biology videos also on there. I've been posting 2 to 3 articles a day basically. So yeah, I've been posting a lot, and it'd be awesome if you guys could check that out. I have a social, thing going, like a social media site type thing going on there and everything like that. So go ahead and check it out if you are into that sort of thing, or just check it out just because. Alright, so before we make this video 40 minutes long by talking too much, let's go ahead and get started. So the first one I'm going to talk about is the gene gun. The gene gun is basically used to inject DNA into a cell. So it injects DNA into a cell. Alright, so that's basically what it does. It's a gun, it points at the cell and it injects the DNA into it. That's basically about it. Alright, and then the next one, automated protein synthesis. So basically what this is going to do is just, it's basically what you think it would do. Automated protein synthesis, you would think it would make proteins, right? That's what it does basically. It'll take the DNA, you can insert DNA into the machine, and then it'll make proteins out of it. So basically that's what it is. So it makes proteins from DNA. Basically that's what it is. Alright, and then electroporation. Electroporation is basically going to be where they actually will put electricity into the cell, and what they'll do is it'll make like little pores in the cell, so they could actually use like a gene gun, for example. The gene gun, you can actually use a gene gun to insert the DNA into a cell. So basically that's what it is. So it's gonna, using electricity, we can make holes or pores in the cell. Yeah, so actually in a little bit I'll be watching the Sony press conference. And I'll try to have an article out by today. I have to make 4 articles today because of Microsoft, Ubisoft, EA, and then now it's Sony. I've already done Microsoft, but I have 3 more to go. So a lot of work ahead of me. So bear with me when I'm not posting that many videos. You know, I don't know if I'll be able to post my Let's Play video on Wednesday because I do have stuff to do again. So I don't know. We'll have to see. We'll have to see. So yeah. All right. Now the next one, cell fusion. Cell fusion is basically when they take two haploid cells and they fuse them together, and you can actually restrict what chromosomes are kept. So basically it's like fusing of cells. Yay, that's basically what it is. Okay. And then automated DNA synthesis. So automated DNA synthesis is basically it just makes DNA strands. I mean, I don't know how else to explain that. It just makes DNA strands based on what you need. Basically, what it's going to be used for mainly is it's going to be used for like making copies of DNA. So, you know, for example, in like a forensics lab or something like that, if you have a blood stain, you're not going to directly get the DNA from that blood strain. You would rather, you know, make copies of the DNA so you could, you know, if you were to make a mistake, you would still have more trials to, you know, there'd be a cushion between making a mistake and then, you know, having it cost. Basically, that's what it does. It just makes DNA proteins. DNA molecules. Sorry, I said proteins. And then there's the polymerase. Polymerase chain reaction, or the PCR. And so basically what the PCR is, it's basically like the automated DNA synthesis, but you can make anything with that, you know what I mean? You can make any polymer with that. So I could start with, you know, a fatty acid or something like that, and then go up to like a glycoprotein or you know, I don't know, something like a cholesterol or something like that, with just a fatty acid. Or I could start with an amino acid and go up to a protein. Basically that's what it's used for, right? And then the next two, we're almost done. So X-ray crystallography. So X-ray crystallography is basically what it's like an X-ray for like really small molecules and stuff. Basically what happens is that you beam electrons into that molecule, and certain parts of the molecule will actually hold the electrons and the other parts won't, and that's just basically dependent on, you know, where the structure is. So basically if we had like an X like this, and this is actually how they found out the structure of a chromosome, the DNA. I should just draw an X. Okay, yeah, this is how they figured out the structure of a DNA molecule. So what they did is they just, they shot electrons at it, and basically every other place the electrons would miss, you know, except for these. And here, you know, then you could figure out, oh, okay, it's an X, or it's a thing, you know. Basically shoots electrons and finds shape. And we are at the last one, so this is really quick video. Protein structural analysis. Protein structural analysis. Basically what this is it's going to be using like it's going to be like an MRI machine. Basically it's going to use like a magnet to find structures of proteins. Now obviously, you know, parts of the protein will actually be negative and positive, and they'll have north sides, south sides. And so basically what the magnet can do is figure out, you know, the overall structure of the protein by using those. So it's going to be uses magnets. Oh. I should just erase that, honestly. me get a bigger eraser. Sorry. Okay. So basically it's going to use magnets. Magnets to find out the structure of a protein. So uses, I forgot to write that. Uses magnets to find the structure of a protein. So that's basically what it does. It doesn't do much else. You know, and we actually, you know, us as human beings, I can say, use this to, you know, make our lives easier. Because, you know, for example, if we didn't have like the polymerase chain reaction or the automated DNA synthesis or something like that, we wouldn't be able to, you know, we'd have like a crime suspect or something like that. And what would happen is that if we messed up on the one DNA, you know, we had, and messing up is actually fairly easy. You know, you have to be very, very precise with DNA and gel electrophoresis and stuff like that. You have to be pretty precise. And so, like, what will actually happen is that you won't get accurate data, and you've lost your DNA. You know, now you have no evidence, and you can't use it for anything. You've lost your opportunity, and you could get fired. So this basically saves us by having multiple copies of the DNA. So basically, it's like, you know, you're writing an essay, handwriting an essay on a sheet of paper, okay, and it has to be exactly that sheet of paper. And if you mess up, you know, you can't erase it because you're writing in pen, and you can't scratch it out. Basically, you have to throw it all away. Now, what basically this is going to do is it's going to, you know, you're going to write a line, you're going to write like a paragraph, and you're going to copy it. And then, you know, basically you don't, it's not that big of a problem if you actually mess up. Now, I mean, that's like not that accurate of a definition or example, but, you know, you get the point. Alright, so I guess I can end the video. Basically, all you need to know is all of this stuff. Just basic understanding will be good enough. And you'll need to know how they'll be used in the, you know, how they can be used at least in the research labs and stuff like that. And that's about it. And I guess I'll leave you at that, and I will see you guys on the next one.